Hello everyone. My presentation on the individual and combined effects of heat stress and Eimeria maxima infection on the ileum function and tissue morphology in meat-type chicken. Eimeria parasite eats poultry producers' money, causing in USA about $127 million as annual losses. Heat-stressed bird is unable to eat or grow and hates its life. Also, heat stress causes about $165 million as annual losses. Coccidiosis is a parasitic disease caused by epicomplexum parasite of genus Eimeria, invades the intestinal mucosa and during its life cycle going through two asexual merogeny stages and one sexual gametogeny stage. Typically, the high degree of tissue damage and immune responses were reported during 5, 6, 7 days post-infection. There are 5 highly pathogenic Eimeria species: Acervulina, Maxima, Bernotti, Nicatrix, and Tenella, infect the commercial chickens, causing high morbidity and mortality. Eimeria maxima invades the middle jejunum and ileum, damaging the enterocytes containing digestive enzymes and nutrient transporters that impacts the nutrients digestibility and the nutrient absorption. High ambient temperature above the broiler's thermoneutral comfortable zone causes heat stress, leading to decreasing in feed intake, growth reduction, and increasing in feed conversion ratio. Heat stress and Eimeria maxima infection are coincident stressors. Therefore, we hypothesized that heat stress and Eimeria maxima, either individually or combined, would significantly impact the broiler's performance, as well as the ileum tissue function and morphology, including the ileal nutrients digestibility, the expression of ileal nutrients transporters, and the ileal tissue histological architecture. Experimental design: there were 4 treatment groups: thermoneutral control, thermoneutral infected, heat stress control, heat stress infected. We had ROS 708 fed on standard grower diet containing 0.2% titanium oxide. Each group consisted of 6 cages, each of 10 birds. There were 2 temperature levels: the thermoneutral temperature at 20 degrees Celsius and heat stress at 35 degrees Celsius. Also, there were 2 infection levels. The control groups received distilled water and the infected groups received 200,000 Eimeria maxima sporulocystis per bird. Birds were allocated and infected at 2 weeks old, and the study has been terminated at 14 days post-infection. Methods: performance parameters were body weight, feed intake, and cloacal temperature. At day 6 post-infection, ileal tissue and contents were sampled. Ileal contents were used to detect the nutrient concentration using HPLC. Ileal tissue was processed for RT-qPCR to detect the expression level of the nutrient transporter genes, the enterocytes gene markers, and tissue oxygen genes. Another segment of ileal tissue sliced and stained with H&E to measure the villus height, crypt depth, and calculate villus crypt ratio. All the results were statistically analyzed using SAS software with 2-way ANOVA model and 2-case HSD post-hoc test. P-value at less than or equal to 0.05 considered as significant. We had at least 5 biological replicates and technical triplets. The apparent ileal digestibility was calculated using this formula. Results: both heat stress groups, heat stress control and heat stress infected, showed significant increase in cloacal temperature, indicating the successfulness of heat treatment. All treatment groups showed growth reduction compared with thermoneutral control group. However, thermoneutral infected and heat stress infected, both infected groups expressed similar growth reduction. The heat stress infected ate less. Taken together, heat stress infected had better feed conversion ratio compared with thermoneutral infected group. Surprisingly, only thermoneutral infected group showed severe reduction in the crude protein and crude fat digestibility. We investigated the expression of the ileal hexose transporters located on the apical membrane and basolateral membrane of the enterocyte and responsible for the absorption of sugar molecules from the intestinal lumen and transport these sugar molecules to and from bloodstream. Similarly, thermoneutral infected group showed the lowest expression level of all sugar transporters except for GLUT1. Also, we investigated the expression of ileal fatty acid transporters, fatty acid transporter protein 1, fatty acid binding protein 1, 2, and 6 that are localized on the apical membrane of the enterocyte, binding the fatty acids and transfer these fatty acid molecules to the enterocytic cytoplasm. Similarly, the thermoneutral infected group showed the lowest expression level for the fatty acid binding proteins, but not for the fatty acid transport protein 1. We also investigated the ileal peptide transporters located on the apical membrane of the enterocytes that are PEP1, PEP2, and peptide histidine transporter 1, and responsible for absorption of di and tripeptides. Peptide histidine transporter also expressed on the endosomal membrane. Likewise, thermoneutral infected group had the lowest expression level of PEP1, PEP2, but not peptide histidine transporter. Opposite to our hypothesis, the results showed that chickens under heat stress and Eimeria maxima infection had higher ileal digestibility and better expression profile of ileal neurotransmitters than Eimeria maxima infected chicken under thermoneutral condition. Therefore, we had generated new sub-hypotheses that heat stress improves the enterocytic function and or prevents the enterocytic damage induced by Eimeria maxima infection. To test our sub-hypothesis, we browsed the literature to select gene markers specific for absorptive enterocytes that are highly expressed in the villus absorptive enterocytes versus the other enterocytic cells. And we selected these genes: acyl-CoA synthetase long chain family member 5, intestinal alkaline phosphatase, and sodium glucose transporter that was already investigated with the sugars transporter. Likewise, the previous results, thermoneutral infected group showed the lowest expression level of the absorptive enterocytes gene markers compared with the other treatment groups, indicating the absorptive enterocytic damage or dysfunction. The histological picture supported that. Here, thermoneutral infected group had shorter villus height than the heat stress infected group. Furthermore, the thermoneutral infected group had the largest value of crepe depth compared with the other treatment groups, indicating the highest degree of inflammation and crepe cells hyperplasia in the thermoneutral infected group. Taken together, thermoneutral infected group had the lowest villus script ratio value. Knowing that the oxidative damage contributes to the intestinal injury besides the Eimeria proliferation, we investigated the expression of two major oxidative genes, CYBB, which is a part of NADPH oxidase 2, and inducible nitric oxide synthetase, that are highly expressed by heterophils and macrophages during the immune response against Eimeria to produce reactive oxygen species and nitric oxide species. Surprisingly, thermoneutral infected group showed 7-fold increase in the expression of INOS. Also, thermoneutral infected showed 1.8-fold increase in the expression of CYBB gene. Conclusion. Even though Eimeria maxima and heat stress are independent stressors in broiler chickens, their combined effects are not synergistic. However, all the treatments affected the chicken's performance. The Eimeria maxima infected broilers under heat stress showed improved ileal digestibility and nutrient transporters expression than the Eimeria maxima infected chickens under thermoneutral conditions. Together with the ileal tissue morphology and the expression of oxidative genes suggested that heat stress curtailed the pathogenic effects of the Eimeria maxima infection and suppressed the parasite-induced enterocytic oxidative damage. Thank you.
Thank you, Mr. Ghareeb, for this very interesting presentation. I was wondering if you've considered the possibility that under heat stress, you have an increased blood flow due to vasodilation to your intestines, hence explaining some of the changes that you've observed.
Hello, everyone. If we consider the increase in the blood flow, we have to think about the increasing the inflammatory inflammatory cells infiltration, and still I think it's not correlated, because my last slide showed that the oxidative species produced by inflammatory cells at the heat stress group is much lower, or even at the same level of the control uninfected. So we can suggest the blood flow has nothing to do with the inflammation degree in case of Eimeria infection. You may have higher blood flow, but still it doesn't mean higher inflammation or higher migration of inflammatory cells.
Another question from the audience. Could the increased nutrient digestibility have any relationship to more nutrients required for maintenance to counter Eimeria and heat stress?
Again, please, I didn't get the question.
Well, if you look, you can click on Q&A at the bottom if you want to see it. But the question is, could the increased nutrient digestibility have any relationship to more nutrients required for maintenance to counter Eimeria and heat stress?
This is one of the suggested points. Bird's body can invest or can try to utilize more nutrients essential for regenerating the mucosal cells or for immune response, and this as a compensatory mechanism. But still, I think, according to my study and according to our previous publication, it was at the same track of this research. we found the effect of Eimeria maxima, the damage effect is higher than the compensatory effect or compensatory response. So if the highest damage, specifically at 6 days, 4, 5, 6 days post-infection, this extreme damage and wipe up all the enterocytes. So even the bird wants to compensate, to express more transporters and to absorb more nutrients. The bird, first of all, the bird has to regenerate like other epithelial lining, intact enterocytes, and then express the nutrients on this newly generated enterocytes. So like the first effect, the highest effector is the tissue damage. Then the expression comes after this, after fixing this damage.
Okay. Well, thank you very much for your explanations and congratulations on your presentation.